Good afternoon. I think the Cozy committee did a really good job by putting all the language model based applications at the end. So I don't want to do a lot of talking, especially in that part because I think everybody wants to go home. So this work is mainly done by two PhD students in my lab, Paul and Suresh. I have a collaborator here, and then Michael, who has helped us tremendously. He is in the acknowledgements. I'm going to show that. So my lab is recently, for last 7-8 years, we've been focusing on predicting post-translational modification sites. So as you know, the central dogma of biology is like you have DNA, RNA is translated to protein. But after that, a lot of modifications actually happen. It looks like there are more than 400 different types of modifications. And, because of these modifications, the proteome repertoire just becomes big. And they're known to be involved in so many diseases and all those things. So it's important to work on those post-translational modifications. The particular type of post-translational modifications we are working or I'm explaining in this particular project is called S-nitrosylation. So what actually happens is these post-translational modifications target only specific residues. So in this case, it targets cysteine C residues. I'll make it really short. Again, there are experimental identification approaches like mass spectrometry and all those things, which is good because without that we won't be able to have a training data set for our purpose. But what we want to do is develop complementary computational approaches, to identify post-translational modification sites. So we started working on PTM prediction around 2014, 2015. Back then, what we normally did is, let's say you want to predict a particular site C at location 50 of a certain protein. If you look at that site C, and if you look at site C in another position, if you assign any physicochemical features, because in order to pass something to machine learning models, this has to be numerical features. Any this C, even though they are at different positions, would just get the same characterization. So just to make it specific, what people do in the field is just create a window around that site, and normally same number of residues on the left and right. And so now you have a window. So if you're creating a window around site 50, and if you're creating a window around site 100, at least there's some differences, and you would use these different physicochemical properties like amino acid index, hydrophobicity, all those indices, all those characterization, people have done over hundreds of years. And then finally, you pass that to a machine learning model, and then you predict whether that particular site is, yes or no. So, but with the advent of language models, and especially protein language models, thanks to Roche Lab and Facebook and other places, now we have this incredible uh, kind of apparatus where a lot of information is distilled in these language models. And basically, what we're doing in our lab is kind of going away from those handcrafted feature extraction and really kind of using the power of these language models to encode protein sequences in order to predict post-translational modification sites. So our approach, we call it PLMS site, protein language model S-nitrosylation site. So what we actually do is we use actually a stacked generalization approach where we have embedding coming from a protein sequence. I have another figure I'll explain it, and then we still also create a window, and for that window we use kind of Keras's embedding layer, and somehow do intermediate fusion to get to a final output. So again, we use this data set. I'm not go into details because I didn't create it. We use some existing data sets, but three things that are really important for any machine learning approach is the data set, the encoding part, and the deep learning machine learning apparatus. So one of the struggle in the field, and probably a lot of field, as I've been talking, is how do you really get that negative set, right? So I think there was a talk this morning where they use positive on-label learning, but again, as we know, positive on-label learning is not as good as positive negative learning. So somehow, majority of us in the field define this problem as a positive negative learning. Positive examples, you can get it from the experimental data, but how do you define a negative site? And it's just a little bit challenging. So normally what we do is, let's say for a protein ABC, site 50C is a positive site. So what we do is we just do approximation, right? We somehow have to start somewhere. So we take another C, because S-nitrosylation only occurs in C, from the same protein which has not been characterized as positive by experimentalists. We take that as negative. And if you do that, normally what happens is you'll have more negative sites than positive sites. Again, you can use cost-sensitive learning, for imbalanced learning. Normally, that doesn't work well, because maybe these are not truly negatives. So what we don't want to lose is the positive sites. So we normally use undersampling to balance, our negative sites with the number of positive sites. So once we have the data set, then the next important stage is how do you actually encode your protein sequence. So here, unlike some of the other, tasks, here the task is not protein level prediction, but it's like site level prediction. We are interrogating whether that particular site is yes or no. So with the help of Michael, because like these language models sometimes are just a little bit intimidating, right? We want to really use it properly, because like there's CLS, there's so many tokens, and thank you, Michael. So what we basically do is we use Proti5, and again, I don't have to explain about Proti5, because like we did a good job by having all these language model talks in one session. But essentially, the input is the sequence from which this site, positive or negative site, is coming. And what Proti5 gives you is it gives you 1024 features per site. And for this work, what we just did is, let's say, we are interrogating whether a site 50, C in this particular protein sequence is yes or no. We just get that 1024 features. So that's how we encode, using Proti5. The other is basically we create a window around their site, and we use Keras's embedding layer. It's just supervised embedding layer. And again, there are some parameters that needed to be fixed. For example, what is the vocabulary size, output dimension, and input length. But you can do cross validation and then figure out these parameters. So once you have that, basically what you do is actually do a intermediate feature level fusion. So for a particular site, you get the 1024 features, and then basically this is going towards here. And then also you create a window around it, and then you use supervised embedding layer. So basically, again, there have been, you have to make so many decisions. This is just one decision we make based on, some cross validation. Basically, what we have is, 16 from here, 4 from here, and then we concatenated those 20, output, and then, go to the final output layer. So these are individual, MCC, Matthews correlation coefficient of individual approaches. The embedding, has pretty good MCC, and then Proti5 has, 0.293. And when we integrate those, basically do the intermediate level fusion, we have MCC of 0.34. So if we compare it against other approaches, we do pretty well. I mean, not great, but at least, the most important thing is now you don't have to do those handcrafted feature extraction, because you don't know what to use. Of course, you can do feature selection, use gene index in random forest and all those things, but this works great. So, also the tools are available at our website. Again, we have a bunch of them now. LM sucksite for succession prediction, phosphorite for phosphorylation prediction. This is the S-nitrosylation. We are working on OGLACNAC. We are trying to attack as many PTMs as possible. Also, GitHub link is available. And then thanks to NSF and thanks to Michael.
Thank you, Dukka. There's someone very keen to ask a question, so I'm sure we have time.
Thanks. I don't know if it's more of an observation than a question, but data set generation is difficult. Since you're only dealing with cysteines, have you thought about using the Euclidean distance between cysteines as a means of separating? It makes it a little bit circular, but you can basically separate cysteines which are annotated as being post-translationally modified, and then take the cysteines which are furthest in Euclidean space to those cysteines as like the negative sample instead of randomly or bootstrapping from the negative set. Yeah.
Yeah, so I think that's a good point. We have tried similar things, but again, have not really found a good approach. And if you have some ideas, we can definitely talk. At least in phosphorylation, what we did is, so we have collaborators who actually do mass spectrometry. They did mass spectry on cladding. So they were saying that if the two sites are close, so for example, if they are less than 5 amino acids apart, maybe you should not take that as negative. So we've been trying to use some insights from experimentalists to figure out or read out some of the positive, but negative sites. I think that's a really great suggestion.